Bismillah ar-Rahman ar-Rahim. I welcome you all in this video. Today we are going to discuss about the Phylum Porifera. But before beginning to talk about Phylum Porifera, the first foremost thing we should learn is the origin of multicellular organisms on Earth. So conceptually, it is stated that the multicellular organisms originate on Earth around 800 million years ago. Now there are two hypotheses. The first foremost hypothesis says, according to the colonial hypothesis, it says that the unicellular organisms are unicellular cells live together and they form a colony. So the level of organization begins from this service. And the second concept, which was a quite recent one in the terms of terminology, that is co-inositial hypothesis. It was earlier called as syncytial hypothesis. It says that a cell divides its nuclei into multiple copies but doesn't retain its cytoplasm towards cytokinesis. So the cytokinesis concept was blocked. It was actually the multiple cells which were formed together, and the organisms of multicellular origin originates using this hypothesis. Anyway, without further ado, let's proceed for Phylum Porifera. So this is the basal level of organization, and over here it is generally called as spawns. So asymmetrical in shape, as you can see, the spawns are asymmetrical or superficially they have a radial symmetry. Three types of cells are identified inside them: pinacocytes, mesenchymal cells, and conecocytes. They have a central cavity from where a series of branching chambers through which the water circulates. They don't have any tissues or organs. So just a basal level of cellular division is happening inside them. Now, the phylum Porifera is further divided into these four classes: calcaria, which is basically the sponges, if the sponge particles are made up of calcium carbonates, and hexactinellidea, it is made up of silica. Desmosponges, over here the sponge contents are made up of desmospongens or spongins proteins, and homoscleromorphs. Now these homoscleromorphs are anatomically simple and crusting in forms. So these are just the names for to remember, and their salient features are already elaborated over here in this slide. Now coming back to the topic, that there are three kinds of cells: pinacocytes, mesenchyme cells, and conecocytes. Now if you look at this image, the pinacocytes are those outer region cells which are mainly considered for carrying or holding food particles on the top of them. So they contain certain flagella. Now these flagellas contain actually hold any moving food particle on it, and then what it goes, it is being engulfed as trapped food particle inside the cell, carried through the food vacuole. From food vacuole, this essential component is being digested and passed on to mesenchyme. So if there are no cells in the mesenchyme, that is called as mesoglia, and then the process is further initiated towards conecocytes. Now the feeding pattern of sponge is starting from the leftmost side, S-cones. What this S-cone is showing is a blue arrow. Now if we look at this arrow, the S-cones are what are trying to do. Water enters from these small pores, they are called ostias, and then move out from osculum, followed by the more complex network which is cycons. Now cycons have certain convolutions inside them, so in-current canals are present and the water again enters, but the surface area of this sponge is slightly more. Then a more complex structure is leucons. Again the water enters using the same osteos, but followed by a certain in-current canals. They emerge into an exurent canal and the water moves out from osculum. So both S-con, C-con and leucons, water moves out from osculum and enters through small pores or osteocytes. This water channeling is actually been facilitated under the influence of light. So if there is a broad daylight, the water movement is high, and if there is no light, the water channels for osteos are closed. So that's the level of what you can say control of sponge for the water flow. Okay, and the reproduction is being facilitated by gynecocytes and amoeboid cells. So gynecocytes turn into sperms and amoeboid turns into eggs, and this fertilization also occurs in an open atmosphere. Thank you very much for your time and attention. If you have any questions, this basic level of Phylum Porifera introduction will help you a lot. And how we could use spawns? Spawns are used in our daily life. If you are observing your door wall, your walls inside the room, sponge can be used as a paint source, as a roller in that regard. Thank you.